DNA replication starts in the middle of the chromosome. Let's look at what happens on the end. So as the replication fork is approaching the very end of the chromosome, so we see that one of the two strands, as the replication fork moves, one of the two strands reveals the 3' end. The 3' end is what we're looking for. Recall ATP, the triphosphate is on the 5'. The 5' wants to go in the 3'. So this is all good. One of the strands reveals the 3', and every time as the replication fork moves, the polymerase just follows it right along, no problem. The other side reveals a 5', and the 5' doesn't work. So the replication process has to put down a primer of RNA. The RNA gives a 3' handle, which you need. So the result of all this is that the replication is always backward on the lagging strand. When the replication fork reaches the end, one side, the DNA polymerase just runs right off the end of the strand, and there's no problems with it. The other strand is the lagging strand, and the polymerization is always going backward. So there's a primer is put down, and it polymerizes up to the previous primer, and then another primer is put down, polymerizes up to the previous primer, and then the last primer is put down. The last primer gives a nice 3' end, polymerizes up to the previous primer, but when the primer is chewed off, it reveals a 5' end. And the 5' end doesn't work for DNA replication. And the result of this is that the last part of the DNA never gets replicated. So the result of all this is that the chromosomes get shorter and shorter and shorter. So you look at this and you say, well, there must be some kind of a trick. And indeed, there is a trick. The ends of the chromosomes are called telomeres. Telomeres are, it varies from group of organisms to group of organisms, but in mammals, the telomeres are TTAGGG. So it's TTAGGG, TTAGGG. The telomeres, the ends of the chromosomes, have a particular function. They protect the ends. But more importantly, as the chromosomes get shorter and shorter, it's the telomeres, which are designed for this, that are getting shorter. So the telomeres are designed for this. This is part of their job. There's an enzyme. It's a RNA protein complex that's designed to lengthen the telomeres. The way it works is that the RNA, it has RNA in it. The RNA forms a recognition site for the TGA-GGG. It binds on, finds the recognition site, and adds another cycle. It adds another TTA-GGC, and then it moves over and adds another one, and it moves over and adds another one, and moves over and adds another one until the telomeres get out to where they're supposed to be. The telomerases are particularly active in the germline cells, which is in the production of sperm and eggs. The telomerases are pumping the telomeres out to where they need to be because there's going to be a lot of cell division going on. I look at molecular biology, and sometimes I think that there's just a lot of idiosyncrasies of the way things work. DNA replication can only work one way, and one of the strands is backward. That just seems kind of odd to me. And then you get to the point that the DNA replication doesn't do the whole thing, and the chromosomes keep getting shorter and shorter on both ends. But the more I look at this, the more I think there's a sort of a beauty and a harmony to it. This is part of the cycle of the cell, and part of the cycle of the tissue, and part of the cycle of the organism. I don't think it's too extreme to say that this is maybe where the circle of life begins. I don't know. But it sort of does make sense. If the genome is a book and the chromosomes are the chapters vary in size. They kind of grow and shrink according to cycles which are only now beginning to be understood. So what does all this mean to us? Well, let's go straight to mammals. An adult mammal there's not a whole lot of cell division going on. The idea of the embryo is that the cells divide and they divide and then they divide, and when the organism becomes an adult, you don't want a lot of cell division. You want the cells to kind of go to sleep on a mitosis level. You don't want a lot of mitosis. The cells are genetically active, but there's not a lot of mitosis going on, not a lot of cell division going on. In a mammal, cell division tends to be things like cancer, like tumors. There's pretty clear evidence that tumors are a result of faulty control of the telomeres, faulty control of the telomerases. There's evidence that the telomeres have something to do with aging. They look at old people and they look at young people and they realize that the young people have bigger chromosomes than the old people. Again, I don't see how this could be anything other than part of the circle of life.